Thank you, Jane, for the introduction, and thank you to the organisers for inviting me here this morning to share some of the work, some of the research we're doing to address some of the challenges around pain in sheep. So the work I'm presenting today represents the combined efforts of a large team of researchers at the University of Adelaide, with support at different stages from the ARC, MLA, the University of Adelaide's Davies Livestock Research Centre, and the SA Sheep Industry Fund for the sheep work I'm talking about. And there'll also be some cattle stuff in there supported by the US Department of Agriculture in collaboration with Kansas State University. Now, because we like to use big, hard to pronounce words wherever possible, we call ourselves the Neuroimmunopharmacology Lab. But basically what that means is we research the interactions between our nervous and immune systems in health and disease, how we can measure those interactions and how we can modify them with treatments to improve health, well-being and performance. And so, of course, that's quite relevant to addressing challenges of pain both in humans and in livestock. Our team is led by Professor Mark Hutchinson, pictured there and somewhere over there. He's lucky enough to be in Amsterdam at the moment at a pain conference, so I'll be doing my best to fill his shoes this morning. To give a bit of an idea of the perspective I'm speaking from today, my background is in precision medicine research. So that is really working out what we can measure, usually something in the blood for me, that helps us either predict or measure someone's response to a treatment. So, for example, analgesia. And so in the past, I've done that mostly in humans, but now I'm applying that to livestock. And so most of my research days are actually spent at a lab bench or in front of a computer trying to extract molecular data from blood to see if it can tell us something useful. So whilst I've spent a few hours out and around the sheep yards and around the marking cradle out at Roseworthy and a few hours in the boning room at Abattoir, I don't claim to have an intimate understanding of the sheep industry. But what I do base my work on in the lab is it has a basis in that sheep experience pain, okay? And that is in response to procedures like tail docking and castration, also during lambing, as well as due to the natural mishaps and maladies and hazards of life as a sheep. And at the very least, that is acute pain. But if sheep are anything like humans and other mammals, then a proportion will also experience persistent pain, especially if that acute pain isn't well managed. And so in order to address wellbeing concerns around pain, we need to be able to measure that pain, to be able to answer questions like, are you in pain now? How much pain are you in, and have you had pain in the past? And that measurement enables us to develop and demonstrate the effectiveness of pain mitigation strategies. It enables us to provide support of our claims of pain mitigation and also support our ongoing evaluation of best practice and provide evidence-based recommendations. So a simple example of that was the assessment of pain behaviours following mulesing that enabled the demonstration of analgesic effectiveness and thus the evidence supporting the current recommendations for multimodal analgesia to be used during lamb marking. And so really what I'm talking about today, the goal is not just about measuring pain, but really what we're talking about is the ability to measure, the ability to provide reliable objective evidence of effective pain mitigation and perhaps even demonstrate a life lived free from pain, or at least free from persistent pain. And so that measurement initially might be just in the context of research trials to demonstrate analgesic efficacy, test whether one approach is better than another, but we can also look beyond that to potentially put tools in the hands of producers to enable them to identify, monitor and better manage pain in their sheep, and importantly provide objective evidence of effective best practice pain management in their flocks. And a critical component of this is working in partnership with industry to bring those solutions to drive improvement on the basis that the risk-benefit ratio is in favour of being proactive as an industry in addressing pain in sheep, especially when you weigh that against the significant downside risk of doing nothing and potentially getting stuck with answers to questions around pain that are no longer acceptable, at least to certain markets or perhaps more broadly in future. So how are we going about this? So the first stage in our research program is to expand our pain measurement toolkit in sheep. Because pain is complex and multidimensional, we need different tools to assess the different dimensions of pain. And I'm not going to stand here and tell you that your sheep can't tell you when they're in pain through their behaviour, but as a prey species, they have a habit of hiding their pain rather than showing signs of weakness to a potential predator. And so what that means is the absence of pain behaviour in sheep does not necessarily mean the absence of pain. So a major part of what we're doing is to add those objective measures of pain, so there's hard numbers to go with those traditional subjective assessments like behavioural scoring. In addition to that, we're looking to expand our measurement tools to not only be able to assess the acute benefits of pain mitigation, but also the longer term benefits, those days, weeks, months, potentially even out to lifelong. Some of the tools we're working on are only ever going to be practical in the context of research trials. But again, we're looking at developing tools, translating those into fieldable, scalable, low-cost tests. So we can deliver a suite of tools for use for selection for different jobs. And one of my particular focuses within our program is to identify candidate blood-based biomarkers of pain and analgesia in sheep. So that is, what can we measure in blood that tells us something of the current pain state or pain history of an animal. And one of the first places we're starting with that is looking at blood, gene expression and regulation changes. And so this is a bit different from the genetics that Alex will be talking about next, which is about DNA sequence. Here we're talking about how much certain genes have been turned on or off and how the levels of those gene products change in the blood in response to pain and analgesic treatment. And we're doing this, we're looking here because we already know in humans and in rodents that we can measure these biomarkers in blood that correspond closely with differences in pain severity. So whether that is difference between individuals or changes in pain severity within an individual over time. In addition to humans and rodents, in cattle now, through our work in collaboration with Kansas State University, we've also identified a number of candidate blood-based biomarkers of pain and analgesia in response to dehorning and castration. So we see multiple examples of biomarkers like this one shown in orange, where the levels go up and down in the blood in close correlation with pain intensity within an animal. And so that's pain intensity as determined by behavioural scoring for multiple observers, which on this figure are shown in grey. And so we see these biomarkers peaking around 12 hours post-procedure and then subsiding over subsequent days. And it's not shown on this figure here, but if we treat with an analgesic, meloxicam, we see a significant reduction in the peak in these biomarkers at 12 hours post-marking. In addition to these, what we call real-time biomarkers, we've also identified other things we can measure in the blood weeks later that still differentiate between our dehorned and castrated cattle and our control cattle. So dehorned and castrated in red up the top here, and our control in grey. And we also see significant reversal with analgesic treatment, in this case with meloxicam, shown in blue and purple there. And so this work is going on to further validate these biomarkers. The US FDA have now come on board and they're supporting us to actually qualify these as endpoints for use by drug sponsors to demonstrate analgesic effectiveness and get the first approvals, the first USDA approvals of the first analgesics for use during dehorning and castration in cattle in the US. And so now we're looking to achieve the same in sheep. So we've taken our first 136 lambs through marking, where they're randomised to either have ring docking, knife docking or no docking of the tail, with and without castration in the boys. And in this particular trial, there's no analgesia. And so we've taken multiple different measurements, including blood samples for biomarker assessment, both before marking and multiple time points out to 12 months post-marking, to see what changes in response to these painful procedures. And so at the moment, we have, over at University of Adelaide, we have freezers full of samples from these sheep that we're working through as we speak. But I do have some recent results from a pilot study conducted in a subset of lambs from that trial that's already providing good proof of concept for our ability to identify blood-based biomarkers of pain in sheep. So this example here, this is just one of the biomarkers we've identified in that pilot study. These data are in U's, and each line on this graph is a different U. And so what this is showing is that how the levels of this particular biomarker change from before marking to completely differentiate between our knife and ring dock lambs in red and green respectively versus our control lambs at both 4 weeks and all the way out to 12 months post-procedure. So that's one biomarker just in U's, but if we take that a step further, take the measurements of a couple of those biomarkers at 12 months and map all of our male and female ewes that were in the pilot study based on these measurements without actually telling the computer which group they belong to. They come out in two nice distinct clusters of sheep, so each point on here is a different lamb. And then once we re-identify those, we see that those groups nicely correspond to our control lambs versus our lambs that underwent marking without any analgesia. So it's really important that I point out now that this doesn't necessarily mean that these sheep are in pain at 12 months post-marking. It's really important. Rather, what it means is there are molecular and cellular changes that occur in response to these marking procedures, and that some of those then persist in the blood so we can then measure them later down the track. So when it's more practical to do so, particularly in a production system. The other great thing about these particular biomarkers we looked in for this pilot study is they can potentially be measured in something as simple as a dried blood spot. And so if these results pan out in the rest of the samples and we pass further validation, not only do we have something we can use in the context of research trials, but we have a test that may ultimately be fieldable, at least for the sampling component. So for me, at least, that's something that's really exciting. In addition to looking at blood gene expression and regulation, we also know in humans and in rodents that we can take the cells out of their blood, put them in a test tube, annoy them with an immune stimulus, and use the differences in the response to that stimulus to differentiate individuals with chronic pain. So we're working on developing the sheep version of that test that can be used in trials at least to do the same thing, to not only identify persistent pain in sheep, but most importantly, we can then use that to test the effectiveness of our candidate drugs we have in mind aimed at blocking that persistent pain. In addition, we know in chronic pain in other species leaves a cellular imprint in the brain and spinal cord. And so at 12 months after marking, we collected all the brains and spinal cords from these lambs, and our PhD student, Charlotte Johnson, and our helpers have been working hard analysing all of those tissues to see what effects of these marking procedures can be identified in those tissues, and perhaps whether tissue collected at abattoir could be used as a marker of or a measure of the lifetime pain history of a sheep. So that as well is a work in progress. We do have an early output from that, which is a bit of a case study for where we had one of our control ewes came in lame during the trial due to a significant lower leg injury out in the field. So that lame ewe was euthanised and we collected their brain and spinal cord. Fortunately for us, that happened towards the end of the trial, and so what Charlotte has been able to do is to compare the brain of this lame ewe to other control ewes in the study. And basically what these sideways blue lollipops tell us is that there are notable differences in cell densities in brain regions involved in pain processing and pain modulation in this lame ewe when compared to our control ewes. So demonstrating we can pick up the effects of at least this kind of serious pain on the sheep brain. So where are we now? As you can see, we're still in those early stages of the development of these biomarkers of pain in sheep, and there's still a lot of validation work to be done. So that's on the measurement side. On the pain mitigation side, thankfully we have those current treatments available that are recommended for use during marking to deal with that acute pain. But as part of our program, we also have Dr. Sam Evans and others who've been working on screening candidates, developing a list of candidate treatments that could potentially be re-indicated at the time of marking that are targeted towards preventing a persistent pain as well. So our next phase of trials, these current and potential new analgesics, they come in as part of those, as additional arms in those trials to both help validate that our biomarkers are actually measuring pain and analgesia, but also to co-develop those new pain mitigation solutions as well. So as of today, to go with our progress in cattle, we have those pilot data I showed you that support the potential for at least venous blood-based biomarkers of pain in sheep. And then as we complete the analyses from the current trial and those subsequent trials that I talked about with analgesics on board, what we're aiming to do is build a more complete map of what a pain-free sheep looks like from a biomarker perspective as compared to sheep who experience pain without analgesia, and then see where our sheep treated with our current recommended best practice during marking sit relative to those. And if sheep are anything like humans, we expect to see variability and analgesic effectiveness. That's expected. and potentially a proportion of sheep with indications of some form of persistent pain. And then ideally with SAM's candidates, we're seeing a reduction in that incidence of persistent pain. So this is all in the context of research trials. We're taking venous blood samples, collecting tissue, that's all well and good. But I've also been asked to speculate a little on what our ability to more reliably measure pain might mean beyond this research trial context. So for some of these blood-based biomarkers, there's a potential for us to convert those into fieldable, scalable, low-cost tests, so something as simple as a dry blood spot, and having built the corresponding pain map based on those biomarkers, provide the opportunity for producers to measure their own sheep, their own flock, to see where they sit on this pain map. So perhaps that's something analogous to a genetic flock profile, or maybe that's something targeted more towards individual sheep, maybe individual carcasses. And maybe there's benefit in doing that, to be able to say, hey, I use best practice pain management in my sheep, and here is the proof. And maybe that provides advantages in or access to certain markets in the future. The other thing, you might just be able to take a pain audit of the flock, identify areas for potential improvement, and see whether your changes in practice are actually having a positive impact. So with these blood-based biomarkers, we can measure them in live sheep, but as well there's the potential for meat sheep to take measurements, including tissue, during processing as well. What else might the ability to more reliably measure pain in sheep open the door to? Well, our primary driver for addressing pain in sheep is one of well-being, but obviously if we can demonstrate production benefits, that's going to help things along as well. So it'll be interesting to see whether once we're able to more reliably measure pain and analgesic response, particularly at the individual level, whether that makes it easy to demonstrate some of those production benefits, particularly long-term benefits of effective pain mitigation. And then the last one here is, we know in humans that your risk of developing chronic pain is sort of roughly 30 to 60% heritable. So once we can reliably diagnose persistent pain in sheep, perhaps that provides an option to select for animals that are at lower risk of developing a persistent pain as well. So to sum up, it's clearly still early days in the development of these objective pain assessments in sheep. And there's a lot of challenges to overcome scientifically before we get to these stages, these tools beyond the research context. But most importantly, we're not just working on the measurement of pain, we're co-developing those pain mitigation strategies as well, so they're ready to go no matter what we identify. And we're doing this in partnership with industry because, the scientists, the lab nerds like myself, we're very focused on identifying these biomarkers, validating those biomarkers in new treatments. And as you can see, we get excited about what we're learning along the way, and we can get really excited about where the science could take us. But we need those reality checks along the way with industry to make sure we're staying on track towards solutions that can actually be implemented for positive benefit. So with that, I'll just thank you once again for inviting me here this morning to share some of our story, starting to tackle the challenge of pain in sheep with the support of the Sheep Industry Fund. Thank you.
Thank you, Dan. Really interesting science, and I think we all need to be aware of where our animals, what they're feeling, and what we're doing, because that's exactly what our consumer is wanting. So I've got a question here.
Dan, Terry Sims, Sheep Central. It looks like there's no research on pain in regards to milking. You're doing it on tail docking.
Yeah, at the moment it's tail docking castration.
And at the moment, so has somebody made a decision not to look at the pain associated with milking?
Initially, that would have been the decision to not do mulesing initially. Not the industry. Well, I don't know. So that would have been Mark's initial conversations, and so I wasn't part of those. I don't know, Jane, whether you would have.
I think the initial
decision.
Sheep that they looked at were unmulesed. So so they've been following along in that line. Yeah. So I think once they get the biomarkers and stuff in place, it'll progress. The whole thing will progress.
So you think that there'll be further work on mulesing, or do you just think they'll be able to apply it to mulesing?
I don't mean to be rude, Terry, but I am no scientist.
So the idea would be that you could apply it to mulesing as well. So there would have been some practical considerations around the ethics of mulesing our research flock that didn't need mulesing for that initial trial as well. So another thing I was going to say there, but it's just dropped out of my mind. Sorry.
So I've got some more questions here. Yeah, thank you.
Dan, you had that graph saying that the elevated levels at 12 months may not necessarily indicate pain. Could that have any reflection on meat quality, pH, etc.?
No idea. No idea. Yeah. Yeah.
So there was
I guess I can say once we get these tools and able to use them on a large scale, collection and abattoir, and you start collecting that data at the same time, that's when you might be able to start to find that. Yeah. Yeah.
So there's some more questions at the back. Yeah, right up the back.
Yeah, it's a tough thing to do. You need really big numbers to do that. So a big part of the development of those tools that can be used at scale, say in abattoirs, so we can actually capture all that data together, and then you can start answering some of those questions. But from these small research trials, there's so many other factors that go into determining the overall performance, I guess, of an animal that, with these size trials, you're not going to pick that benefit up. And I'm not going to make any promises today about a production benefit. Yeah.
Practically, there's a lot of people that may use multiple pain relief products at their market.
Mm-hmm.
So like a tricycline and a analgesic. Are you looking at that as well?
Yeah, that next phase, that'll be part of it. Those analgesics now come in. I mean, with mulesing, those initial trials, the thing with mulesing is that the pain from that is a lot easier to measure through behaviour. Definitely the indications with that are the combination of the NSAIDs plus your local anaesthetics does provide a benefit. Then we'll be able to see pain from tail docking and mulesing is perhaps a bit harder it is harder to pick up. It's much harder to pick up with tools like behavioural assessment. And so they're the kind of questions we'll be able to answer is there benefit in using both for just tail docking castration.
In your approach, how do you differentiate between pain and inflammatory process, or how does that work?
Yeah. That's my biggest scientific challenge is how we actually do that. Is actually generating confidence that what we're measuring, particularly these blood-based biomarkers, that they're actually measuring pain and not injury, not wound healing, things like that. So I could talk for 10 minutes on that challenge. There are multiple ways we're tackling that, but there's always going to be a level of uncertainty around there with pain because you don't know what the ground truth is. That's one of the things. So if it responds to analgesia, that's a good sign. And some things have a really clear biological link. So the stuff we're looking at spinal cord is, you can be pretty confident that is a pain process, not something else. So yeah, you have to come at it with a lot of different angles. Yeah.
So I just want to start off by firstly acknowledging the carcass comp last night. What a great result. And for a couple of MLA board directors and AWI board directors back in 2003, we told you so, because that's when we started the Merino Validation Project. And I said to them at the start that when we put eating quality into a carcass comp, merinos will win. Well, I thoroughly enjoyed last night. And I have sent a text message this morning. So, with that little bit in mind, my job today is to cover off a little bit of genomics. I firstly want to acknowledge Peter Bradley, Julius Vanderwerth, Daniel Brown, Rob Banks, and those sort of people who provided a few slides. Matt McDonough from the Wagyu Association provided some in-confidence slides, so I can show you a bit more data around that. I want to give you a bit of an interpretation about where genomics is at the moment and where genomics goes in the future, and then I'll leave the last slide up for your interpretation about where you actually sit. So in terms of sheep, and one thing I missed yesterday was a lovely feel-good day, wasn't it? We felt good about our industry. I'm here to put the reality back into this situation. Right? The lamb industry's got to compete, and we compete every day. We compete on several factors. We compete in both the domestic and the international consumer dollars. Right? We are putting a product into the marketplace that consumers are paying dollars for. They expect fantastic eating quality. They expect welfare. They expect sustainability. If we're not delivering any of those, don't bother selling it in the international market. Competition for land use. In Australia, we compete against cropping, we compete against beef, we compete against cotton, we compete against whatever you want to look at. Right? If we're not competitive lamb producers, putting lamb on the market at a certain rate, we're not in industry. So who here knows their cost of production? 3, 4, 5. Yeah, bloody hope for similar. Righto? Competition for resource use. We're starting to emerge into an industry now where not only are we talking about water, we're talking about methane, we're talking about fertiliser resources, we're talking about all these other things. Who's benchmarked themselves in terms of kilograms of land per unit of P, per unit of millimetre of rainfall? 1, 2, 3, 4. Yeah. And that. So that's going to become more important. And then we've got competition for the climatic voice. Right, so we heard that very distinctly yesterday. And the sheep industry must have that climatic voice. So if the one thing that comes out of this presentation is you put as much pressure on AWI and MLA, and if you're in New Zealand, Beef and Lamb New Zealand, to fast track methane research to get a methane breeding value into the marketplace as quickly as possible, you've achieved something for today. Right? Because without a methane breeding value, we won't hit the 2050 targets. All those ameliorates are going to be great in the short term. They're not going to deliver those long-term impacts that we actually need. So you need to think about that, and I'll come back to that. So the key facts. Genetic evaluation now, if you didn't know it, is heavily influenced by genomic predictions. Genomic tools are available for most livestock species. So when it comes to competition, if you don't use genomic tools, and beef is using genomic tools, and cotton is using genomic tools, and cropping is using genomic tools, how competitive do you think you'll be in 5 years' time? Not very competitive. So it's probably time to invest, and for commercial producers, you will be able to access very good tools in the not too distant future. Again, another learning experience. Go and talk to Daniel Brown and Peter and those sort of people about some of the new tools that are coming out, and I'll show you the impacts of those in a minute. So life before genomics, and I deliberately put this slide up in black and white. Relationships through pedigree, difference in performance came from contemporary groups. Difference came from other areas and relative performance. We spent hours talking to breeders to use genetic linkages between flocks. We spent hours trying to convince merino stud breeders that they should use strands from other sources. And that was a real good experience, wasn't it? And I'm still doing that today. We spoke about accuracy of progeny testing, and we spoke about access from trace from direct measurements only. Righto? And for the person, if you want to see that, that's me many years ago with my father and mother trying to work out which one of us is dyslexic when we're collecting information. And let's just say I lost the battle. But we collected all of this information and we put it all in, and that gave us a fantastic base. And we put all that information into a database, and with relatives we were able to produce breeding values from contemporary performance and maternal half-sibs and paternal half-sibs, everything like that. It worked hunky-dory, and we made fantastic genetic progress. Right? We were able to generate real changes. So for an impact of genetics, extra dollars, 1.67 kilograms of lamb, about $210 million added annually to Australia's bottom line just from genetics in carcass weight. So genomics is allowing that trend to continue. So what have we done? So we've introduced a simple little tool that measures genomics. It measures sometimes up to 50K, which is 50,000 SNPs, sometimes up to 100K, which is 100,000 SNPs. And when we have genotypes, instead of putting the relationship or the pedigree relationship between animals, we fit the genomic relationship. So we fit the relationship with all other animals that have genes in common. So it changes the equation pretty quickly. So the problem is when you have genomic information, you fit that genomic relationship. When you don't have genomic relationship, you fit the pedigree relationship. So you make these pedigree orphans. They're not as accurate, they don't have the integrity of the information, and they don't compete. So I talk to ram breeders and other people about genomics, and I say, what proportion of the flock are you measuring? And and they say 2%, where's that? Yes, he's not in the audience, is he? So he's changed his mind. So when you do that, you actually find that you're measuring only 1 or 2 animals and they outcompete everything else because they're so much more accurate, right? And they have so much more other data. So again, if you're a ram breeder, you need to start doing much more in that area. The real challenge is that all of this information is built on about 15 years of information coming out of the sheep's RC information nucleus, have big reference populations both in sheep and beef and dairy, traditionally industry funded, and that's been great. The challenge in 2 years' time is who's going to fund those reference populations, because MLA and industry have decided they need to do some other investments, which is great, and there's some new tools coming out of those investments. But reference populations need to be funded by industry. So you need to make the choice now of how do you get as much information into those reference populations as you can. Because if you don't, other industry players will start developing their own reference populations, be they large corporate supply chains or artificial breeding companies, and all of those are happening in the dairy and the beef industry right as we speak. So in the dairy industry, I now sit on DataGene as a board member. There's about 30 different breeding values internationally that are coming into the international marketplace from different reference populations, all on similar bulls, right? And that is the real challenge. It used to be expensive to create a reference population. It is now no longer that expensive to do so. So those are changing, and because of that, we're getting these predictions emerging on high use size, which are different. And who is going to validate those genomic predictions in the future? Right? At the moment, industry validates everything, both here in New Zealand. In 3 or 4 years' time, it could be very, very different. So that's a question for you and industry to look at. So as I said, we've got single step, we use all the information, we accept all those tools, we put the G in the relationship, much higher accuracy, and I'll show you that in a minute, and we get an increase of opportunities to look at very difficult and hard to measure traits, such as methane. The issues, we accept the genomics works, right? So the challenge with genomics is it pulls animals which are related further apart, and I'll show that in a minute. But you get this increased accuracy. So as my wife says, when my daughter and I, we have a small Angus stud, we do a little bit of genomics. She said, oh, you're playing snakes and ladders again, right? When you get the genomic test back on it, some animals go up and some animals go down, and it's a bit like playing snakes and ladders. And it cost me $50 an animal to do so, which she thinks is a great idea. But the challenge of that is how do you actually understand how that's working, because that's coming in. So there's less reliance on pedigree. There's less, and it relies on this reference population. And the big challenge is you can be a breeder and put in an animal with its genotype and be related to animals you would never have considered you've been related to in the past. And they influence your breeding value. Right? So you're no longer an orphan in the industry. Every time you genotype an animal, there will be a proportion of genes which will be common to every other animal in the breed. And that's going to change in the not too distant future, because sheep genetics has the merino genetic evaluation at the moment, a terminal genetic evaluation and a maternal genetic evaluation. They're going to combine the terminal and maternal into one hit and create a really interesting reference population that is going to really revolutionise how we select prime lamb size. Right? I didn't say maternal or terminal, I said prime lamb size. So again, the reference population allows us to do that. You get much higher accuracy, so for the boffins in the room, you get about a 30 to 40% improvement in accuracy by having breeding values, genomic breeding values. The big challenge is we can select animals at much younger ages. So literally the day an animal's born, a lamb or a calf is born, you can take a TSU sample, you can send it away, and within 6 weeks' time, you'll have a full genetic profile of that animal back. More accurate than a sire that had 15 progeny 10 years ago. Right? More accurate than a sire that had 15 progeny 10 years ago. So it changes when I start to make some selection decisions. So in terminal sire breeders, if you're not using ram lambs, you're out of the game. In maternal sire breeders, if you're not using ram lambs, you're out of the game. In merino breeders, if you're not using ram lambs, you're out of the game. Right? It's as simple as that. He's changing the world. And again, we're starting to see the ability to select young ewes at much younger ages. Who here started using IVF? Not yet, they should be. Who here started to use sex semen? Not yet, you should be. And I'll come back to that and show you an example about that in a minute. So, fundamental principle, got population of animals, you do all the accuracy. That's just making sure that there's some integrity in this talk and not some other areas. But importantly, you don't have to be an industry body or breed society to own the reference population. Supply chains exist in pork, semen companies in dairy, large corporates in beef and sheep. That could be the future if industry doesn't make very informed decisions in the next 2 years. So, why am I excited by that? That's a ram that's Anderson Pole. Thanks to Peter for this. Traditionally, half-sibs share about 50% of their genes in common. With genomics, you pull that apart. So you can actually see in this case, the half-sib genomic relationships sit anywhere from 15% out to 35%, and they should be 25%. So we pull genetics apart with genotyping. And the really interesting thing is not all progeny have the same level of relationship with their sire or the dam. Some are more related, some are less related. So the old concept of the pre-protein sire, genomics is actually confirming that's actually has a bit of truth in it. Don't tell the old timers that, but there is a bit of truth in that because there is more relationships coming through from that. And here's an ET case study thanks to Andrew and Jamie Heinrich. And this is where I get excited. So this is a full variation in full ET siblings, and they all were genotyped, and we were able to demonstrate there was at least a 1.5 micron difference. There's like 20% difference in fleece weight, and more importantly, and what I often get excited about, is there was up to 40% difference in worm resistance in full sibs coming out of an ET program. So the old merino breeders who used to do these sib matings and throw a whole heap of sibs out in the paddock and expect them to breed to type, that's crap. Righto? And it's crap because of this. And this is the important part. If you're investing in genetics and genomics, as a ram breeder, you can pull everything apart. So you pull out the animals which are very, very different and you make more genetic progress at a faster rate. So I get really excited by that. And I get really excited by telling merino breeders and terminal sire and maternal sire breeders they should be ripping into ET programs using sex semen because we can pull things apart much more succinctly. And here's an Angus example just on ET siblings, anywhere from down at 0.3 out to 0.75, right? And that's, for people who want to know, that's paratrooper, right? So that's the relationship there. So, genomic prediction tools. We have genomic enhanced ASVVs using both phenotypes and genotypes. We already have flock profile, a great tool. I think there's the smart people in the room. Oh, sorry, the Neogen people in the room. They are now smart flock, I think. Is that right? Something like that. Anyway, they've got that. I know there's Zytogen, there's others there. Flock profile allows you to rank your flock against others. It's based on DNA tests, 20 animals per flock. We'll include condition score and weaning rate in the future, and new MSA indices, right? It's a great tool, right? And in another couple of months, there'll be a flock profile for both maternal and terminal flocks as well. So if you're a commercial breeder and you want to know where you sit in the big bad world, flock profile is for you. $800 plus a little bit for management, those type of things, but it gives you a benchmark. It's like carbon baselines. If you don't know where your baseline is for genetics, don't come whinging to me in 2 years' time when people ask you questions about methane and other traits. So I put methane and genomics in the same boat as doing measurements for carbon. The other big projects emerging is the new genomic breeding values coming out of AGBU and UNE with Julius and Daniel and others. Sheep genetics, sheep metrics, Neogen, Weatherby's and Zytovet. And it's going to take the genomic breeding values out of the single-step run for sheep genetics and allow commercial breeders to access that same information. Maybe at a differential cost, that's up for people to work out, and maybe there'll be a different set of traits. But again, it is a valuable tool, and I'll show you why it's important in a minute. But that's great. So that is coming out, and you need to have that. There's flock profile. Why I get really excited about flock profile is yesterday, that result told me that if I have a flock profile, I can go to a processor in 6 months' time if got a merino flock or terminal sire flock or a composite flock, and tell that processor exactly where my lambs will sit with regards to eating quality and yield. Right, for $800, every commercial breeder with 500 ewes and above should be doing that. It is a no-brainer, right? Now, why I get excited about genomics in the sheep industry is because I dabble in the beef industry as well, and I compete with the beef industry. So sheep competes with beef. Don't get it wrong, we compete, right? We compete in a whole range of different areas. And the problem is, beef has already got all of these tools up available. So Heifer Select, Inherent Select, the Black Blocks, which is a new test going into the north around reproduction rate and fertility. That's Matt McDonough. He wanted his picture put up with Wagyu Feeder Check, Integrity Beef, and the new Breed Select. There's at least 15 different tools emerging in the beef industry and much more in the dairy industry. So they have those tools today, and they are using those tools to outcompete sheep producers because we haven't got the tools yet available. Right? They are making changes at a rapid rate. And if you want to have a look at the future, there's the dairy industry. That's a TSU, that comes out of ST Genetics, a very well-resourced company in the US and now in Australia. That's their vision product, and they're claiming with one genotype, they can do robotic cows, so it gives you an idea about robotic milking. They can do pole, they can do muscle weakness, which is a genetic inherent trait. They do eco-feed, which has got a methane trait and a sustainability trait in it already. They can do all the genomic breeding values coming out of 2 international genetic evaluations. They put it all there, they tell you that, and then they also tell you the sex even to go with it, and then that makes all the decisions. Pretty simple, isn't it? It costs you $35. How do you compete against that unless you have the right tools? Right? It is very, very simple. So, I've been lucky enough to play with one of those tools, Heifer Select, in the last couple of months. 4 herds pay pretty good money. They pay very good money for bulls. One's a Texas herd, one's a Paraway. I don't mind saying this there, Burramuka and Twynham. We've got another herd which is a Team Tamania herd, and a 4th herd which is based on Arthur Cox's really good Angus herd in the northern half of Australia, New South Wales. All of them are really good cattle producers, all run about over 800 breeders. All of them have been Angus breeders for the last 20 years. All of them pay big more money. All of them think their cattle are the best. And then I went through and genotyped all their heifers, and boy, did I have some fun. So, within any trait, herd 1, 2 or 3, there was animals which are in the top 5% or animals in the bottom 5% for most traits. And they had no idea of the variation within their herds. Right? And we were able to pull that out. In terms of marbling, herd number 2, they thought they were really good marbling, and in fact they had more than 60% of their herd below breed average for marbling because of their selection process. Herd number 3, they had challenges around mature cow size and a few other things. And more importantly, they all had really good animals that they didn't understand. When we actually sat down, some really progressive producers will tell me what the real result of this whole thing was. What do you reckon the first thing that happens when beef breeders make selection decisions on yearling heifers? What do you get rid of? Not one of them had a heifer out of a heifer in their top 100. So they're culling out on phenotype. Every one of them loved being cattle enthusiasts. They loved watching cattle. So they were all selecting big mature size heifers, and their mature cow weights were going through the roof because of that. They didn't have an idea of that. Because of that, their calving ease was going downhill at a rapid rate of knots. So Heifer Select allowed them to do that. That's the first look at Heifer Select. There's all the traits you can look at. There's your new indexes that are coming out of Abacus Bio in New Zealand. And there's the range that exists within each of the herds. Huge range within each of the herds. The challenge is that particular herd, when they took out their top 100 heifers and they put them into the AI program, we were able to see that they now had new heifers coming in that were out of heifers, and they were selecting animals which were more moderate in size and had much more productivity for the long run. And we've just got some calving results, and I'll explain that. The other big thing, and I was excited by Mark Ferguson yesterday saying he could do $4.50 a lamb or $4 a lamb yesterday, I think was what he quoted. Yeah, you should go back to him and tell him you want it for $3.50, but he's not in the room, so that's good. But here's a really good example. Here's one particular property, all the different sires have been blacked out, but we were able, because of heifer select, to work out which sire had the most heifers that were in the contemporary group that were kept. Huge variation there. Which sire had the best results to AI? So some sires had 100% conception to AI, other sires had 20% conception to AI, and then some sires had 60% empties in their heifers that were retained, and other sires had none. All based on size. So that all came out of genomics. All 4 people this year are doing genomics of their own free will with my guidance. Right? And that is a big challenge. So that's exactly what you could do with the new tool that Daniel and others are developing, is the capacity to do exactly that within the sheep flock. And there's the lifetime value. We worked out that it probably sits at about $1,000 a heifer. 5 minutes? Great. Now I want to talk on feeder check for the Wagyu industry. I do a little bit with the branded beef competition, but it's important. So someone said yesterday that lamb is the new Wagyu, or Wagyu is the new lamb. I think I read that correctly. You've got a hell of a long way to go to keep the top end of these guys. There are 2 people I know in the lamb industry that are anywhere near it. These Wagyu guys have spent a lot of money looking at feedlot gain, carcass weight, P8, fat, eye muscle, obviously marble score. And marble score drives everything in the Wagyu industry. They've now got a genetic test that sits out there that they can screen all of the commercial animals going into a feedlot and have these genomic predictions before they enter the feedlot. Right, so they are now custom feeding animals based on their genomic profiles. Wouldn't that be great to do that with the lamb industry? Because we have to. It's not an if, we have to compete. There's some of the variation that they get from the top to the bottom. Great tools that are available. And this is the thing that I get excited about. This is their customised dashboard that Matt allowed me to show you today. No one should take a picture, Terry, but you can. Picture one. So that's a whole range of animals going into a feedlot. They've all been genotyped. They've all been run through feeder check, and they're their rankings, and they are separating those animals based on whether they go into a 400-day grain-fed, a 300-day grain-fed, or whether they be rejected. Right? And they reckon the value of that is about $400 to $500 at the point of feedlot entry. So there's great tools, and you get these spider graphs and everything like that. You benchmark all your size, you benchmark all your dams, and you basically change the profile of your herd overnight. Right? How many sheep producers would love to have that tool? Righto. Daniel, stick your hand up. That guy is the guy you need to talk to. He looks a little bit older now than he once did, and wears glasses. But Daniel Brown is the guy you need to see at morning tea, because that's the type of tool that they're developing for the future. And I think Peter told me they'll have it hopefully available within the next 12 months, Daniel. That's right. Wouldn't it be great to have that? And there's another one where we've benchmarked a whole heap of size. In this case, that particular herd I know very well. I've done a little bit of work with them. We brought in 3 outside size, benchmarked them, never used them again. Right? So you make these rapid turnovers. So that's the big thing that breeders will have to get used to with genomics. You no longer have a love affair with your rams, righto? And I know there's breeders in the room that do, and I'm looking at Norm and Chad, they have these love affair with these rams. I want to use them every year for 5 years, once only, and get hard. Righto? That's the challenge. Because you have to turn over genetics. So there's some really important areas. So a note on current pricing. Where do I think the challenges are? Dairy genetics, you're paying about $2,000 of replacement female, and you're getting a genomic test for about $45 to $55. Makes sense. Wagyu feeder check, and remind you that little star should be there as well, so you've got the on-costs of being members of breed societies or access to other select areas as well. Wagyu feeder check, $1,500 to $3,000 per carcass is what they estimate at $45. Heifer select, $1,200 to $1,600, $42. So it all makes sense that you can invest that type of money. Flock profile, as I said before, 500 ewes, $150 a ewe, $75,000 capital value. spending $800 to benchmark yourself is a no-brainer. Every commercial producer in this room today should be down at Zydavet or Weatherby's or Neogen or organising their flock profile. It's as simple as that. Right? And the reason you do that is when that methane breeding value comes out in a couple of years' time, you've already got those profiles in the system and you have that benchmark straight up. If you don't, you will take another 12 to 18 months to get that, and that'll be a waste of time. You have to get into the game. So, what does genomics mean for ram breeders? And how many people breed their own ram? Yeah, ram breeding enthusiasts, I call them. Yeah. So, ram breeders and ram breeding enthusiasts, much more relationships between the genomics and pedigree. Genotype animals get the full benefit of single step, right? If you're a ram breeder, both a serious ram breeder or a ram breeder breeding their own rams, you have to genotype. There is no sense not genotype. Doesn't matter what breed you're in, you have to genotype. Non-pedigreed animals and non-genotype animals, my view, waste of time, right? They don't get the full accuracy, don't have the full spread, don't give me the confidence. Much younger accuracy at younger ages. Makes young sire and young dam, 2 minutes, great, one side to go. Young slides, young dam selection, much more accurate, brings IVF and sex semen in. So any ram breeder in the room who's not using IVF and sex semen in next year, you're out of the game. Simple as that. Right? There is so much good to be used with both of those two technologies. Brings in the next generation of competition. One thing I wanted to go back, I'll just go back quickly because I wanted to say it. The reference populations, right? We've got methane genotypes in Australia, New Zealanders have got methane phenotypes coming out. There is not a single logical reason why the two countries aren't combining methane phenotypes for a combined reference population for the sheep industry to compete against the world. So the industry players that are sitting in the room today that have abilities to help with those decisions, fix the issue this year. You've got this year to fix it. And brings in that next competition. Commercial breeders, breeding values indices can be bought in and benchmarked from anywhere. You can predict and sit where your flock profile. So if you don't know where you sit, right, that's your choice, but don't be in the professional lamb industry or don't be a wool producer if you don't have it. Opportunity to source genomics from multiple sources. So people used to say, oh, I can't use that ram, he hasn't been tested in my environment. Well, that's all crap. Because I can pick a ram from the New England or I can pick a ram from WA or pick a ram from Tasmania and they all have high accuracies and I can get the full suite of traits. So ram breeders can be much more adventurous, so commercial breeders be much more adventurous. Changes how you select your replacement females. I bet you every merino breeder who picks out young ewes, picks out young ewes out of adult ewes that are born in the first cycle and look good. Right? This tool changes how you select animals. It changes your flock profile and changes your ability. Next generation tools are going to be available for novel and future traits, and that's where sustainability and welfare comes in. There's a whole range of areas, and as Dan said, if Dan has a genomic marker in 2 years' time, you better make sure you have some genomics in the kit to use some of those genomic markers. And how long will it be before lambs have to be marketed like Wagyu? Probably 1 or 2 years' time. The best processors in the world, i.e. Rob Davidson and his crew at WAMCO, and Dawson and others, will be doing genomics to work out who has what lambs and when lambs are ready. So my last slide, Madam Chair, is pretty simple. If you're not using genetics and you're not using genomics, you can make your own choice, but don't blame me. Thanks very much.
I'm sure there are going to be a lot of questions in the room, and Terry, I'm going to let the producers have a go first. Firstly, the people standing up the back of the room, there are seats up the front if you'd like to make your way up. Please come and do so now before our next speaker. Questions? Terry.
They're all shy.
Terry, go. No one else has put their hand up.
Wool growers are coming up for wool poll this year. I've just seen results AWI put up where they linked their China spend to supposed increases in sales, yet we find that that in fact is linked to the price of non-wool fibre rather than their spend. So that begs the question, they're going to get a levy figure. Do you think that levy figure should be spent by containing those nucleus clocks, or full foaming, rather than anything else.
The levies, I'll answer that very simply. The levy spend should be spent on novel and new traits that are going to help us be competitive in the future. So we're not spending information on resource blocks around methane, or new sustainability traits, or the new welfare traits that are emerging in the future. Then I think there's some real issues because the rest of industry can't pay that. Running resource blocks for just simple traits, not worth it. Running resource blocks for hard to measure, futuristic, not commercially viable now, but really important in the future, no-brainers for any of the decision makers in the room.
Yep, thank you.
As a commercial grower myself, so second cross land, I'm buying my first cross ewes in, you're talking about doing a flock profile. Absolutely. My ewe base is 250, 300 ewes from wherever I can get ewes every year. So I don't have any two ewe lots of the same. Is it worth it considering my genetic, like the next lot of buying, if it's not from the same breeder, I'm sort of buying a new genetic lot of eggs here, if that makes sense.
So two ways I'll answer that. With flock profile, you're going to have to do 20 animals. So I reckon that market is about 500 ewes per nucleus set, right? With Daniel's mutual coming up in the next year, you might be able to do 10 or 15 ewes from each of those different components and benchmark those different components and work out which one you really should be buying.
Yeah,
Right, so
that's always been my thought.
the bloke who's follically challenged over there, sitting next, and Peter's next to him. So they're the two that you've got to follow today, right? Find out where that new tool is available, because that's exactly what I do. If you're above 500 ewes, you should be doing that. More importantly, the people you're buying your ewes off, if they don't have a flock profile and can't show you their flock profile, that's the question to ask.
One more question. Last question, sorry, up the back. Yep.
Alex, I agree with that nuclear reference population thing with those hard to measure nutrients. But as a levy payer, both with AWI and MLA, I can't think of a better spend than even those like long term. We need that reference population. Now, possibly the terminals and things like ongoing supervised things, there's very good for that can probably fill the void. When I put my radar hat on, there's not that same sort of similar thing. Where else can the levy payers' money better spent after a talk like that? On ensuring that reference population is there, it's independent, it just goes on into the future.
Yeah, so it's a really good point. I mean, when a number of us in the room, we saw the concept about the reference population back in the original SRC, setting up reference populations for eating quality and those types of things. Right? We did that with the belief that in 10 or 15 years' time, these traits are going to be really important for the land industry. I still think you should do that and think about what traits are in the future. The animals that are in that reference population, there's no reason why you can't get the rest of the phenotypes that are there and keep the momentum of the reference population going. There is, and I'll challenge anybody to come up with a benefit cost analysis which is more competitive than investment into a long-term reference population of our hard to measure traits that have an impact on industry. Right? Because I do benefit cost analysis. There's no other ones that, there are others that compete, but there's no other ones. Particularly if you've got sustainability, welfare, climatic voice and those type of things in the future. If we don't have genetic information to answer that climatic voice in the future, don't bother being involved in that discussion.
Sure.
They can. Yeah, that's my answer. But being an ex-R&D,
I think we're going to have to leave this conversation.
I'll give you one thing. Being an ex-R&D manager, right, there are always new innovations that are challenging for the public with money. That's the challenge. Your challenge is to convince the people that they should be investing.
Please go and see Alex in the SBA lounge and have a chat with him at smoko time. So we really need to keep on track. Please put your hands together for Alex. Sensational presentation. Yours is up there. Can we have the next presentation, please? This man doesn't need any introduction. Jason Trompf is going to talk to us this morning around new rules for engagement with ewe lambs. So thank you, Jason. And I'm tackling you if you go over time.
Thanks very much. Well, I appreciate that. And we sort of thought this session, if we we're going to spare a bit of time, it would be for Borley, because a lot of you haven't had much exposure to the great man in some time. So it's nice to see you get that exchange with him, which is great. I'm going to talk to you about a project supported by MLA. It's been going for about 5 years, called More Lambs from You Lambs. All the collaborators are up there, in particular Andrew Thompson at Murdoch Uni, but also a shout out to Lynn McCubel and the Bessel program over 10 years ago on farms like James Petty, he's in the room. You know, we contributed data and we've added to it for many years, and really the purpose of this project, collaborated with New Zealand, was to pinpoint where we had gaps in understanding, and it was less so at the front end and more so at the back end, but I'll talk about that, about reducing wastage, and and so there's a whole team of us that have been committed to ULAMs for a long time. And what I want to say is I reckon we need to press rewind, because we promoted this to industry and get in and give it a go and do it opportunistically. I call bullshit today. This is not something you just choose to do off the cuff without great preparation and great discipline. And we need to press rewind a bit, because there's a lot of people using practices like overjoining ULAMs. And I do want to say something right up front, and I don't think we've done this well also in the adult ewe area. The messages I'm sending today are for commercial people. The seed stock farmers in the audience, I want you to unlisten to the best practice side of it, because I want you to keep testing genes. Okay, you're in a bit more control of your systems than the average commercial farm. I'm going to talk about minimum cut-offs for joining and other things you can do with cribbing and joining later and all this stuff. I don't want you doing that. We need you to find ewes that can lamb it back on their birthday, and they can do it at a whole range of weights and find animals that push the boundaries. These messages are really centered for commercial people. So where there's variation, there's opportunity, and we've really got to up our ante on attention to detail. So Kimball Curtis, who Borley and I are using a lot, did a 500 survey, 500 producers across Australia, and you can see that there's people doing this across a range of breeds. It's about 20% that do it regularly, and another 10 that sort of flick in and out of it. So you've kind of got a third of producers giving it a go. You can see the average marking rate, soak up the average ewe mortality rate, and think about an average and where the range might sit around that. And have a look at down the bottom. This is why we don't have good data on what's really going on with ewe lambs, particularly the bottom row. A lot of them are lambed communally with other age groups, and we didn't pick up on the wastage. I'm not going to spend much time on the barriers today, but I think there are some legitimate barriers out there across different breed types and different production systems. And I think one of the ones the meat farms have really got to weigh up, if you're focused on meat production per hectare, this ewe lamb thing, or a merino farm that's really passionate about that as well, you really got to weigh up your investment in this versus running a few extra adults and where the sweet spot sits. And that's an area where we'll sharpen up our messages going forward. So I'm going to spend a moment on this slide, but this is what we've talked to the industry about for 10 years. All these drivers that set potential. We can just about wind this thing up like a 2-bob watch to get whatever outcome you want. The problem is, and I spoke to Sandy McKepper next door, and I said, what do you think of you? And he said, there's an overemphasis on conception. What I actually have at the end of the day in yield is the challenge. So I'll talk a little bit about these things and the extra ones we learnt in our project, but in the time frame, I want to really emphasise the ewe and lamb survival piece. So we know a lot about drivers. I'm going to talk about the top 3 or 4 in a bit of detail in the next few slides. In the middle, I've chucked some of the genetic ones. Tom and others have done great work on understanding, particularly in merino, some of the drivers, and you can see some of the traits there that are favourably related to it. The one I'll give a shout out to is the birth status. What we've learnt is a ewe lamb that is born a twin, or a multiple, if you want to say that, compared to a ewe lamb that is born a single. We don't know how they're reared exactly on commercial farms. She'll deliver the same scanning rate at 5 kilos lower joining weight, the one that is born a multiple. So if you want me to unpack that for you, you get on with selling the singles and sell the weight, and try and get these better end of your twins. Some of them will have been reared as singles, don't die in a ditch over that. And you can get better scanning rates out of those animals. So that's one you can play, and one that ties in, I think, with Bawley's story about not just getting sucked into phenotypes and size. And going forward, I think this message can really live alongside some of the great stuff he was talking about. And down the bottom, you know, there's lots of stuff we know about. They have a shorter, more fickle, less overt estrus cycle, and they're not as precocious in their behaviour. They don't seek out the ram as well. So joining ewe lambs in containment is a good thing. You know, and up your ram percentage closer to 3 than 1, all this stuff we know inside out. The bits I'm going to spend a bit on is this weight thing. So there's zillions of data in here, and shout out to Tomo for the slide, but basically per kilo of joining weight, it's worth 3 to 5% in reproductive rate, or as a farmer we call it our overall scanning rate. One of the things I think you need to understand how responsive your sheep are. It's one of the earlier messages in lifetime wool with adults, per condition score, how responsive. Well, where do you sit in this spectrum? Because it influences the payoff. So I think you need to understand that. The other great paper that Tomo's recently written out of this is, when you look at this slide, it tells you over and above weight, there's this additive impact of body condition score. To the tune of per half a score, it's worth about 20% reproductive rate. But if you're only joining some of your ewe lambs, the use of a combination of scales and condition score only gives you about a 2% improvement, and you work your way all through that, and it's really cool analysis they did in this paper. Basically, you can do it all by weight. If you're only joining a subset of your ewe lambs. This is the next one I want to chat to you about, is another trial that Tomo did, and this is one where we're going to have to repeal a little bit. So I'll be straight out, this was done on Murgippan merinos in Western Australia, and it's all about growth during the joining period. And what the trial found was per 100 grams extra, you pick up 20% in reproductive rate. What's interesting about those sheep, they sit right out the far end on that previous slide on their response per kilo at the point of joining. So one of the gaps that Paul Kenyon from Massey said we need to go after is we don't have enough data on this Trompf. You're going out to do all this work, we've got to find out where it really lies in the population. And it's nowhere near that first story. So what I can show you here, and I just want you to focus on the 45 kilo line each time, and we're cranking up 100 grams of growth rate. This is during the joining period, between rams in to rams out. And the response is less than half of what was found in that original trial. And we've got a lot of sheep in this across a whole heap of breeds. Maternals, merinos, Chevies. So you have to understand where you sit in this responsiveness. You're going to have to do some hard yards. Get someone like Shep over there. He loves getting farmers measuring stuff, learning out their response curve, use your EIDs, work with your scanner, not that hard. And you'll bank the result. And you need to bank the result because if you go and look at the dollars and cents side of things, it changes the message we gave to industry. What I want to say to you is this growing them fast during joining is not a get out of jail free card. And I'll say to you, it seldomly pays. John Young tells me we need $9 a kilo for lamb and $200 a tonne for supplement to get that to pay. The other part I don't like about the message is the guts in the middle of this slide. We can grow them 100 grams a day during joining and get the 8% gain. I want you to do the 100 grams a day for the 35 days beforehand, get them 3 or 4 kilos heavier. That'll give you, depending on your responsiveness, and I've gone for the middle number of 4 per kilo, that gives you 14. So we nearly doubled the response. But the kicker's in the bottom line. What the next piece of work I'm going to talk to you about is every 5 kilos that you'll lower at the start, you've got to gain 10 to 15 kilos extra during pregnancy to offset the risk associated between being light at the start. So you must do your hard yards early. What that bottom line is talking about what I'm about to talk about. And it's the trade-off with animals that are too light at joining, that die either themselves or their lambs die. And don't just imagine that they're sort of disappearing up a rabbit warren. They still go on the full pregnancy, a lot of them, unless there's been any abortion store and the wastage comes in. And that's what this piece of work for the first, and thanks to the patients at MLA, because we had to spend the first 2 or 3 years trying to find out what was really happening. And what we found was, on a lot of sheep across a lot of breeds, I took all that out, and you can see on 20,000 fetuses, we marking 13,000 lambs. So that's 1 in every 3 that you can see is disappearing between the pissing contest of scanning. Sure, as Ken Solly tells us, you don't start high, you can't end high. But if you go on Twitter and listen to the music about all this scanning rate on ewe lambs, there'll be no one on their lamb marking rate. Because either they don't know it or they don't want to tell you about it. And it's because of this issue. And then you've got the sleeper of the ewe mortality. So we've really got to get focused on this wastage piece. So the first thing we did was look at this data set and said, what can we find out within the data set, and also with some brilliant analysis that Tomo have done with the Cashmore Oakley data set. Now this isn't with treatment influenced trials, which I'm about to go to. This is somewhere where Baldy might slap me on the finger and say, you know, take it easy with what you're saying here, because we're just looking at all this profile that we've collected, and we're trying to work out where we go to next. And what we found was, with joining weight, it was indicating a 10% improvement in both singles and twins in the lamb survival area. Faster, higher weight gain in late pregnancy was also associated with much better lamb survival. So we needed to then go and do the treatment responses on that, the hard yards on the trials. The extra thing we also did was a massive trial on the use of Regnuin as an antioxidant to help with these hypoxic births. The same in humans, same in sheep. The first birth is the longest, the slowest birth. You have the risk of oxygen deprivation during that birth. You have partial brain damage. The finest of fine motor skills is for that little neonate to suckle the teeth. And we wanted to find out, because back on the earlier slide, which I failed to say, when you saw all that low, like a 69% marking rate in singles, you're all sitting in the room thinking, oh, they're jacking that lamb. That wasn't the story. There wasn't a lot of dystopia for pulling lamb. I was thinking there might be this secondary dystopia going on due to oxygen deprivation. So we went after that. We're going to publish a paper on it. Overall, there doesn't look to be much, but we're fortunate enough to have some flocks I've already mentioned that we're measuring birth weight, and we're going to see if there's any relationship between birth weight of the lamb and the use of Regnuin and so on, and then in survival. But I'm going to focus on the treatment trials in a second. We had 2 or 3 heads up from a paper that Tomo had written on the Cashmore Oakley data set. The biggest driver of the birth weight of a lamb out of ewe lambs is joining weight. You have to get your joining weights up. That when she sets the whole plane to transfer nutrient from her to this lamb. It's the foundation of the development of the lamb. What's killing lambs out of ewe lambs is too low a birth weight. And this paranoia about putting low birth weight rams and putting them in Jenny Craig paddock like we've had to do with, I would say to you, imbalanced cattle that you can't grow appropriately. The only way you jack a calf out of it is putting them in Jenny Craig. Get your genetics right, exactly what Paulie just said. We can unravel that to fix it. We don't need Jenny Craig to get lambs out of ewe lambs either. We actually need to keep them growing. So what he also found was a strong relationship between joining weight and the weaning weight of the lamb. I'm going to pick up a bit of time here. The other one that he didn't pick up on, but I did, having a bit of a background in the seed stock world, I said, Jono, you're taking laminin scores. So we then redid some analysis looking at the ewe mortality area, and we found that there was this link between the lambing scores and the joining weight of the ewe lamb. And this is why we need people like Jono tested genes. So we started to see some info at the seed stock level. We had a hunch out of our data set. And then we went after setting up these nutrition trials with over 3,000 ewes in them. And this is just the overall profile. And so what we then did at scanning, we split these things into high and low nutrition groups on commercial farms. You can imagine how that went without the use of the research stations we have around the country today. Hard yards, because the same farmer's paying a levy, and Trompf is knocking on his door to say, well, we need to just use your farm as an overgrown research centre for 6 or 8 weeks. Yeah, hard. But they know who they are and they put up their hand to help, which is cool. Interesting, you notice at a pre-lambing weight, this is not maternal weight, this is total weight of the ewe and the fetus and whatever wool she's got on her back. You'll see a lot of numbers either side of 60 kilos. Interesting number. So you can imagine in our data set, we've got animals spread all around that. The first thing we had to prove was whether there was a a treatment effect, because we're on commercial farms. So we tick the first box. Between the high and low growth treatments, we had a significant difference in the weight gain of the ewe lamb between scanning and lambing. So we tick that box. Then is when it gets really exciting. In both singles and twins, no difference. What we found was, between the treatment groups, was an 8% improvement in lamb survival. So that's 8% extra lambs marked in singles, 16% extra lambs marked in twins. What was also really exciting is, when we did the analysis, even though the treatment groups were in the second half of pregnancy, we've got a lot of profile data by now. We're 4 years into a 5-year project. We've got all these joining weights, scanning weights, lambing weights, and the analysis tells us that there's no difference with when the weight gain occurs, whether it's between joining and scanning or between scanning and lambing. So in the next slides, I'll talk about total pregnancy weight gain, which simplifies our extension. Now, this is a little bit hard to digest after the night out that a few of you had, and we've got to work out how we extend this. But there's a real interaction between joining weight and pregnancy weight gain. The next half a dozen slides use a traffic light sort of concept. The first one I've thrown you up is a ewe lamb that's scanned with twins. I've got a joining weights on the left, how much weight she gains during pregnancy, and the percentage of those ewes that dies. Never before have we had this data, and it completely changes the messages. The tool we've got for using ewe lambs has to be rewritten to accommodate this, because it promotes overjoining, it promotes get out of jail free by growing them quick during joining, none of which think about this wastage issue. And if you think this number's alarming, wait till I show you the next one on the mortality of lambs. So you can see if they're joined under 45, and Bawley's going to be down the back, and he's going to puke. There was a project called the Maternal Central Progeny Test Project, and it said 45 by 7. I reckon even Pethick with his meat science would have heard about that message, even though what he was doing was more important, yeah? So we always have fun between the meat scientists and the production people. Seriously, there's been a misinterpretation of that message. It was a minimum joining weight. Read the original report. The sheep industry has a big fat problem. They interpret it as a target, unlike the cattle industry where we talk about minimum critical mating weights. I'm not sure what happened with the message. I ring up the farmer and I say, what's your joining weight? You know, what's your target with your ewe lamb? And they don't say to me, it's 45. If she's not 45, I won't put the ram in. They say, oh, the target's 45. And then I ask them the real question and they go from 35 to 60. That's our issue. It's animals, the tail end of the mob shouldn't be going to the ram. And their lambs die like flies as well. Why? Think through it logically. She gets pregnant light. The biggest driver of the lamb's birth weight is her joining weight. The only way you get out of jail is a shitload of pregnancy weight gain. And I can see James Petty doing the figures on that. He's going to sit down with Sandy. And I think what they're going to conclude is less is more. Stop overjoining ewe lambs. Pinpoint those that you're going to join at weaning and do a proper bloody job on them and get 100% lambs marked on the ones you join, rather than overjoining and doing a half-assed job all the way along. Singles. Thought you'd be interested in that. So what do we got here? Impact of joining weight and pregnancy weight gain on the percentage of single lambs that survive. Nice easy slide to interpret because the lamb survival is your marking. And I've made the green above 90, the amber in the 80s, and the red sort of down less than 80. You can keep noticing this 45 plus 15 gets you where you need to be. So what do you know? At day 135, you're doing your pre-lambing treatments. They've got to stand on the scales. If you want success, there needs to be a 6 in front of the number. Because picture the twin when she's doing that. She's going to drop out with all the conceptus and fluid, 10, 12, 15 kilos. She's going to go from 60 back to 45, right? She's got to rear a lamb and then get pregnant again. So the new one that we're going to come up with is this, and if this starts to blow your head off, I accept it first thing in the morning, but it's a terminology called reproductive success, where I say with a ewe lamb, we want her to stay alive and at least rear one lamb. Right? And so what we've done in this slide is bring the ewe and lamb death into one message. Because if you put it to the ram, you're doing it with the intent for certainly her to stay alive and to try and have the ewe of the lamb. So in singles, you can have a look. If you want to get into the 90 zone, it's 45 plus 15. What I would say is you'll start to notice, and there's some people in Western Victoria this year that did the hard work early, by getting them up higher weights earlier, it offsets a bit of that risk for pregnancy weight gain. So I've got some demonstration flocks this year that have some that have joined at 40 and some that have joined up at 50, and we're measuring their pregnancy weight gain to start to demonstrate through a PDS how this actually plays out. Same story with the twins. So I think you start to get the gist. The old 45 boy, I don't know if it's 10 or 20 years later, is back. But it's back as a minimum guillotine weight. All right, gone are the days of this aloof target. It's a minimum. But the other thing that's back is dusting off your scales where you're all focused on measuring the voids, because that's where the cash flow is. I call bullshit for that. No one is using their scales at scanning and pre-landing for their unit. No one. I've done a survey and the number is a big fat zero. So we've got to continue to grow this thing and manage its profile. I'll only quickly do this. It's an old graph, but the numbers are what we got overall. There's more data than this. Ewe lambs are uber sensitive to mismothering. So the two biggest killers of their lambs is low birth weights and mismothering. And they are really, really sensitive to this community issue of how many ewes around them are going to land on the same day. What we told the industry in adults, that per 100 ewes it was worth 1% marking rate in singles and about 4% in twins marking rate. In ewe lambs, it's 3 to 4 times more responsive. I think that makes sense, doesn't it? She's lambing down at 12, 13 months of age. They're more fickle. She needs more privacy. So you've got to think about how you're going to set them up for success. So we've done some work on optimum mob sizes. It depends on your stocking rate. This is done at $6 or $7 a kilo for lamb based on permanent fencing, not with electric fencing. A lower cost intervention will mean you can bring those optimum mob sizes down. You'll see typically the ewe lamb proportion of optimum mob size at the different metrics is about half that of the adults. So another re-education message, sorry Daniel, but we've got to get out there after we did all the hard work with adults. We've got to just tweak things with ewe lambs. I'm only going to do these quick. This was a more recent survey, fairly switched on farmers, heaps of them joining ewe lambs to the tune of about 1,500 a year. They're joining nearly 80%. They joined for 6 weeks. They're all cribbing pretty much and joining 35 days after their birthday, basically. So they're really joined at 8 months, not 7. We need you to keep working on genes to find animals that can do this on their birthday. Join at 7, lamb at 12. We've got all these lambings going, multiple lambings going on farms, and the complexity of what's going on, I think we've got to take stock. So I just wanted to quickly show you, there's a little bit of adoption of minimum critical mating weights in a biased group, but none of them are doing the weighing, as I talked about. And what do you know? There's the results. 1,496 ewes joined. There's your marking rate. Same wastage as I found in the research. And a big fat question around ewe mortality because the data just goes missing because they end up in amongst other sheep. So my key messages are this: you must thoroughly evaluate the pros and cons of joining ewe lamb. Does it suit your genotype, your labour availability, your production system? You cannot do it half-baked. It's not an opportunistic scenario anymore. You've either set up to do it or you're not. You might pull the lever every year, but you either can do it or you can't. Proactively manage these ewes. It's exactly the same message. If you want to dig out bred wool, fed wool for cattle, it says exactly the same thing. You've got to grab these heifers or ewe lambs at weaning, manage them proactively to joining, manage them through the joining period, then their pregnancy, then their recovery. The ultimate success is converting your potential into reality. It's not the pissing contest of scanning rates. And the final couple of messages I got in this is less is more. Work out a way that you can mark 100% on ewes joined. So we go in with a smaller number and do a better job until we perfect it. And to save a question, if you want to say what about all this percentage of adult weight? You talked about these weights of 45 and 60. How does that relate to Mumblebone versus Johnny Kilo? We're going to go back, hopefully, to the adult ewes in all the trials. And there's a couple of farmers in the room that feel sick now because they're going to get a phone call. And unfortunately, Trompf has got to visit them again to weigh and condition score all the adults. We've got a brilliant data set You hear numbers thrown out around the proportion of adult weight, the work's never been done. So it's all lickety split. You've got to go and do the hard yards to find out what the truth is. Is it 60% of adult size of joining? Is it 70? We don't know. So that'll be the next Mark II when we're a year or two down the track. So thank you. And thanks for all the support to the farmers, both in your levy and collaborating with this research, because we can't do it without you. Thank you.
Thanks, Jonathan. I've got about 3 minutes for questions. Yes, thank you.
South East South Australia and Western Districts, it's about to hit wall. If it doesn't rain for the next 2 weeks, it's gone.
Sorry to hit you.
Yeah, we're out of hay, we're out of grain, we're out of money.
Yep.
And we'll have a new land. What do you suggest is our direction going forward to get them back into shape to stay in the business?
Right. I'm going to go back a step first on that. I don't want to cry over my own spilt milk, because I'm in the same boat, right? You need to think about this ewe lamb thing. If you're pushing hard with the reason I did this research is most of the time adults are lambing in the winter and the ewe lambs are early spring. And 5 years ago I saw a type ewe like you got now. And what I saw was the ewe lambs don't grow during pregnancy. And this is what alerted me to chasing this, right? So the current scenario you're in is why I did this research. What we've got to get better at is pulling the parachute way early. So if any of you in the room have ever been to a Tom Hall breakfast, ironically for 10 years he's promoted to his farmers. You might join these ewe lambs, but you might get to the point where it's near scanning or just after scanning, and you don't keep them on your farm. Remember, for most of the systems in Western Victoria, the only dry sheep on the farm is the ewe lamb. They should have pulled the parachute earlier. And if their system's half decent, they'll bounce back to full production within 2 years. So she shouldn't still be there. And the reason that they're limping through is they don't really understand the mire of this data, which is fair enough because you've only just found it. You can't do it up, but you're asking me what are we going to do at this point? The only things you can do is try and have enough nutrition to keep them alive during lambing. Your marking rates are going to be dismal because your pregnancy weight gains are way back. You'll have to wean early and try and get the recovery in that year. That's the only choice. We've got good data on recovery. I don't have time to present it today. You must use this as an elastic band. Now, there'll be people in the room saying, I can't do that. They're the best crop. Just create another crop for next year. Because the moment you put the ram to her, you are making that animal vulnerable. And if you can't deliver, either because of the season or your management, she's better off leaving the farm. I know that sounds really harsh. My farm's in the same circumstance. We sold our ewe lambs back in March. Right, now you'll say, how did I know the season was going to be crap? I didn't know, but we were tight for land base, and I just knew based on this work that I wasn't going to be able to deliver the goods. So I sent them the meat works.
We've got time for one more question. Thank you.
Is it true that a new lamb that lambs as a lamb is more productive throughout her life over
so this So this comes with our great New Zealand collaboration. We're going to do more of this, and Borley's spot on. We've got 2 sheep industries that are going to shrink up their own arsehole if we don't start working together. Excuse the French, but that's a fact. And one little area we've made a start is in this ewe lamb space. And what they've done is a brilliant piece of work on this space. And basically at the end of the day, and repeated again more recently in a recent paper, about 12, and the girl that did that, published that's now working with Tomo and Murdoch, about 12% more productive for the rest of their life than the other animals. Those that you can get, this is the kicker, they've got to not just get pregnant, they've got to rear a lamb. That's what the recent data has shown. So it's reproductive success first time, programming them. Is there a little bit of genes in there? Borley could tease it apart versus the merino maiden work we had that programs them and sets them up for their life. It's a behavioural thing as well. I don't really care in the dust and the detail. I like genetics and management. Reality is you can set them up to be more productive for the rest of their life. Great question. And thanks for your time, and rip into LambEx today and let's have some fun.